Thank you for having me here today. And today I'll be talking about the molecular stratification of triple-negative breast cancers. Over the years, my laboratory has been involved in the analysis and classification of breast tumors, primarily using DNA microarrays and patterns of gene expression. And through these many publications, we've arrived at these 5 different tumor subtypes in a normal breast-like group. And what we can see is that even though we use no knowledge of patient survival or outcomes to derive these genes, these different classes, again based upon patterns of gene expression, have important prognostic implications. And today we'll be focusing on ER-negative disease, and particularly triple-negative disease. And certainly what can be appreciated is that in the absence of therapy, these tumor subtypes tend to have a poor prognosis. And so much of the discussion today will be focused on the predictive markers for these groups because, by definition, they lack ER, PR, and HER2 in general, and therefore there are few treatment options and a great need to develop targeted therapies for these specific subtype of tumors. So, over the years, we have collected a data set of about 470 different breast tumors. And on these tumors, we have clinical ER, PR, and HER2 data and microarray data. So we applied our molecular classification to this and then broke out the group of triple negatives. And what you can see is on the left pie chart, as is appreciated by most, the vast majority of triple negative breast cancers are of the basal-like phenotype, which is shown in red in the pie chart. However, you can also see that all the other molecular subtypes are present. So we have patients from the Her2 Enriched group, Luminal A, Luminal B, and a few of the normal-like. As I will describe later in the talk, one of our newest tumor subtypes that we call the Claudin Low Group, if we bring that group into the discussion, it is also quite numerous within the triple-negative cancers. So within triple-negative breast cancers, the take-home message is that there is still significant biologic heterogeneity, and it is likely that we will need to specifically target this heterogeneity to be effective against triple negatives in general. So let's start with what should be some of the major constituents of the triple-negative breast cancers, and I'm going to start with actually the group, what we call Her2 Enriched. And you'll see why basically some of these are triple negative. So clearly Her2 positive tumors are a clinically important and appreciated group. Clinically, Her2 positive tumors are about 15% of all tumors, or 20%. And the majority of clinically Her2 positive tumors fall into this Her2 enriched subtype. And about 30 to 40% of these are ER positive, and the majority are ER negative. But if we take a blow up and look at the gene expression patterns of HER2 itself, what you can see is that not every tumor in the Her2 enriched subtype is HER2 positive or HER2 amplified. Hence, we've given it the term Her2 enriched as opposed to simply HER2 to signify the fact that not all are HER2 positive. But this is important for this discussion because those tumors that are in the Her2 enriched subtype and are not HER2 positive are triple negative, right? And so there is a biology dictated by HER2 that has some bearing upon the triple negative group. It may be that these tumors have mutation of HER2 in the kinase domain or something like that. We don't know what this activating event is, but nonetheless, these are a constituent of the triple negative group. Now, for HER2 positive tumors, their baseline prognosis is poor. And obviously, a great deal is known about the benefit of trastuzumab, lapatinib benefit, potential chemotherapy implications, and other things like promising agents like TDM1. One of the pressing questions is that for those patients in the Her2 enriched subtype who are triple negative, do these HER2 targeted therapies have any benefit? We don't know, and hopefully we'll figure that out in some clinical studies. Here is a clinical study that we're involved in with the CALGB. I'm on the CALGB breast group. And this is a study focused on HER2 positive patients receiving either trastuzumab, lapatinib, or the combo. It's somewhat unfortunate that these HER2 enriched subtype patients who are triple negative won't get into this study. So we won't actually know from this study, or many like it, where the inclusion criteria is to be HER2 positive, whether they gain a benefit from these HER2 targeted agents. But there certainly is some hint that there are some HER2 negative patients who gain a benefit from HER2 targeted therapies, and it might be those HER2 enriched patients who are triple negative. Moving along, what most people call triple negative cancers, I call basal-like breast cancer. Basal-like breast cancer is 10 to 25% of all tumors, depending on the demographics of your population. And as I showed you earlier, if you look at just triple negative breast cancers, the basals make up about 50 to 75% of the triple negative subtype. From a biologic perspective, we believe that this is that these tumors are derived from a distinct cell type or a distinct developmental stage of mammary epithelial cell development. We called it basal-like many years ago because of the unique expression of cytokeratins 5, 6, or 17, which are typically expressed in the basal epithelial layer of your skin and of your airways. Now, over the years, we and many others have come to discover a lot of important features about the basal-like subtype, including that the vast majority of these tumors are P53 mutated. These tumors are highly proliferative on average. On the bottom of this slide, we have a very nice proliferation index. And no matter how you measure proliferation, Ki67, pCNA, immunohistochemistry, gene expression for proliferation, this group is homogeneously high. And this is in large part due to their lack of RB1 protein functioning, right, which is a critical regulator of the cell cycle, basically can put a brake on the cell cycle. They're RB deficient, P53 deficient, and thus they're likely growing quite quickly. We've also come to learn that they are associated with BRCA1 mutation status, where the majority of BRCA1 mutation carriers, when and if they develop breast cancer, develop basal-like breast cancer. And we believe that this P53 loss, RB loss, and BRCA pathway association is responsible for the high aneuploidy seen in these tumors, where they have a huge number of chromosomal changes, translocations, and losses. This, getting back to the BRCA pathway association, has important implications due to the now targeting of BRCA pathway alterations using PARP inhibitors. Here's some very promising data from a triple negative metastatic basal patient trial where they received a PARP inhibitor and there's a survival benefit. So Dr. Carey will be talking more about this, and so I will move on. Another important question in triple negative or basal-like breast cancers is chemotherapy benefit, and in particular, is there a carboplatin benefit? And so we're addressing this question in this CALGB neoadjuvant trial, where patients will be randomized to get carboplatin or not. And in my mind, the important question here is, we know there's a chemotherapy benefit in this group to many different chemotherapeutics. Is carboplatin special and even more effective in this group than some of the others or not, is the question that we hope to address in this trial. In this trial, the patients are also being randomized to bevacizumab, or an anti-angiogenesis agent. This is a particularly attractive and important question in the triple negatives. And if we look at this graph here, what we have is the average expression of 3 genes, VEGF on the top, ER, and HER2 on the bottom, according to your intrinsic subtype, where going higher on this plot means higher expression and lower is lower. And what you can see is that on average, VEGF expression is highest in the basal-like and HER2 enriched subtypes. And therefore, if the presence of the target is any indicator of benefit, then you would rationally pick the basal-like group. In addition, we've developed a signature of hypoxia, and as you can see on the top right, that 13-gene hypoxia signature is also at its highest level in the basal-like and the Claudin Low Group, again suggesting that angiogenesis inhibitors should be tested within the basal-like and actually the Her2 Enriched and the Claudin Low subtypes. So therapeutically, what are some of the best targets for the basal-like group? Well, their baseline prognosis is poor in the absence of therapy, and so we must treat them. The chemotherapy benefit is high, and I'll show you some data on that, I believe, in the next slide. But I think there are many promising new targets and approaches for this group, including PARP inhibitors, a potential Carboplatin benefit, the angiogenesis inhibitors, and then also a Her1 RAS-MEK pathway inhibitors. We have some preclinical and actually clinical data to suggest that this pathway, the Her1 RAS pathway, is active and important in the basal-like group. The key issue there is how do we effectively target it. So here are some of the data on a chemotherapy benefit according to your intrinsic subtype, which is taken from a poster that we presented at the last San Antonio meeting. And what we did was to look at the pathologic complete response rate according to your intrinsic subtype, combining together 3 different neoadjuvant studies that had similar anthracycline and taxane containing regimens, namely the ACT arm from NSABP-B27, a TFAC neoadjuvant chemotherapy study from the MD Anderson, and then the ACT I-SPY study headed by Laura Esserman. And at the bottom there, we've combined these 3 together, and I think this nicely illustrates what's been shown in many different publications, which is that in these neoadjuvant studies, the Luminal A subtype patients tend to be the most frequent. However, they have the lowest and a very low PCR rate. In this case, it was 7%. The basal-like group has the highest PCR rate, 43%, which is quite high, actually. And the other thing I'd like to point out is that the Her2 Enriched subtype had a PCR rate of, I think, about 38% or 39% in the absence of any trastuzumab, therefore showing that this group is chemotherapy sensitive, and when we add trastuzumab on top of it, it even goes higher. The other thing to point out is that the Luminal B group has a appreciable PCR rate, but it's still rather low. If we do a multivariable test, what this analysis suggests is that the most important thing to know is your intrinsic subtype, and that knowing ER/PR and HER2 status doesn't add on top of knowing your intrinsic subtype in terms of predicting pathologic complete response. So also over the years, we have attempted, and I think successfully attempted, to develop immunohistochemical surrogates for the intrinsic subtypes, such that we can look at rich pathology archives and to expand the breadth of our studies. And so using our immunohistochemical surrogate, we applied this to a unique population-based study from North Carolina that purposely oversampled for African Americans and purposely oversampled for young women with breast cancer. And this was done to address the following question, which is that it was known that African Americans and young women with breast cancer have a lower incidence, but a higher mortality. So why is that? So using the blocks from 1,400 cases from the Carolina Breast Cancer Study, we did our immunohistochemical surrogate and had the following finding, which was first published in JAMA in 2006 and then in Breast Cancer Research and Treatment in 2008, which is basically that the frequency of this poor outcome basal-like subtype changes according to age and race, where it is at its lowest frequency in postmenopausal Caucasians, then goes higher in premenopausal Caucasians, then goes higher still in postmenopausal African Americans, and it is at its highest level, 27% approximately, in the premenopausal African Americans. This finding in and of itself can explain some of the racial outcome disparity differences because the African Americans tend to be getting more of the aggressive and poor outcome breast cancer. Interestingly, the Luminal A subtype, this good prognosis tumor subtype, has the reverse finding. Its frequency is at its relative lowest in the young African Americans and at its highest in the postmenopausal Caucasians. This study has been taken a step farther by Fumiyola-Patti and colleagues, where we looked at Africans. And what you can see there is that the frequency of triple negative cancers even goes higher in Africans, suggesting, and then if we actually look at, say, a Japanese population, the frequency of the triple negative in the basals is lowest there. So there must be environmental and or genetic factors that have an influence on the frequency of the subtypes according to different populations. And so one means of getting at what might be some of the etiologic causes of these frequency differences was to look at some of the risk factors from the Carolina breast cancer study. And we could see that there were a number of risk factors that actually had opposite effects according to your tumor subtype. Importantly, features like having multiple children and a younger age at first birth are protective of luminal A disease, but predisposing to basal-like cancer. Other features like a high waist-to-hip ratio, which is a measure of obesity, are predisposing for both types of ER-positive and ER-negative cancers. But it was quite stunning to us to see that some of these well-known risk factors, like having multiple children, might actually be predisposing to one subtype and protective of another. So clearly additional studies are needed, but I think this again suggests that basal-like breast cancer is a unique and distinct disease from the ER-positive disease and needs to be treated as such. Now our newest tumor group is what we call the Claudin Low subtype, and we named it this because the defining feature of this group as by gene expression is the low expression of many of the Claudin genes, including 3, 4, and 7. And the Claudins are involved in epithelial cell tight junctions. Another epithelial cell interaction protein is E-cadherin. And so what we can see is that these Claudin low tumors lack these cell-cell junction proteins. The Claudin low tumors are also triple negatives, and so you can think of these as another subtype of triple negatives in addition to the basal-like group. And as you can see by this clustering analysis, the Claudin lows are also somewhat similar to the basals, but distinct. Other important features of the Claudin lows is that they almost always have an intense immune cell infiltrate, and they also have stem cell features and features of epithelial to mesenchymal transition. So here's the histology of 4 Claudin low tumors, which was of interest to us because of this low expression of E-cadherin hinted that they might be lobular, although I think you can see that most of these tumors do not have lobular features, and they're generally characterized by a high tumor grade, particularly little differentiation. And again, they very often have an intense immune cell infiltrate. Now, one of the reasons that we believe they have features of, in this case, tumor-initiating cells or stem cells came through this study with Jenny Chang, Jeff Rosen, and colleagues and Chad Creighton at Baylor University, where in a very elegant study, they performed fluorescence-activated cell sorting on primary human breast tumors to isolate the stem cell fraction or the tumor-initiating cell fraction by isolating the CD44 positive, CD24 negative fraction. They then compared this tumor-initiating cell fraction to the other fractions and identified what they call a tumor-initiating cell signature, which was highly enriched for genes that have mesenchymal features, including ZEB1, twist, snail, and things like that. What's relevant for this talk was that we then took this signature of tumor-initiating cells and compared it to each of the intrinsic subtypes. And what you can see from this waterfall type plot is that it is really only the Claudin Low tumors that were statistically enriched for this tumor initiating cell signature, and it was pretty much every Claudin Low tumor was enriched, whereas only a few other samples from the other groups were. So this is one piece of evidence that I'll show to you today, suggesting that the Claudin Low tumors are somewhat naturally enriched for tumor initiating cells or stem cells. And so this group may offer us a unique opportunity to be able to study tumor initiating cells and stem cells through the examination of the unique properties of this subtype. So therapeutically, what you can see from the Kaplan-Meier plot here is that in the absence of therapy, the Claudin Low tumors have a poor prognosis, similar to the prognosis of Basal-like tumors and Her2 enriched subtype in the absence of therapy, and therefore we must treat. They are, on average, triple negative, and therefore this limits the treatment options. There is some chemotherapy benefit in this group, and so it is likely they will get chemotherapeutics, but we need to do better than that. An important question is, are PARP inhibitors going to be effective on this group? Because they too may be linked to BRCA1 pathway alterations. And then certainly an intense area of study is to target cancer stem cells. If we can do that, then it will be highly relevant to this group. So in closing, and sort of from a biologic perspective, what we believe is that the tumor subtypes, the intrinsic subtypes, are reflective of arrest at different stages of epithelial cell development, with the Claudin Low group representing the most primitive tumors that are the most similar to the mammary stem cell. And then the next step on the pathway is what is sometimes referred to as the luminal progenitor, and this is the Basal-like phenotype. And somehow, if you have a BRCA1 pathway mutation, you get stuck at this step. We then postulate that the next step would be what's called the late luminal progenitor, and that this step mimics what we see in the Her2 enriched subtype, which is basically to lose the basal characteristics and to begin to gain the luminal characteristics. And then at the bottom, we have the most differentiated groups, which are the luminal A's and the luminal B's. So in closing, I'd like to thank my laboratory and my many collaborators around the world and the country. Thank you.